One last point I want to make before we're done with SDS page gel and can move on to other things, and that's what I want to talk about is how I can estimate molecular weight. Okay. So if I plot the molecular weight, and here I'm going to plot the mobility, or RF. This is the top of the well. This is where our marker dye ended up. And our proteins moved a certain distance down here. And we're going to define the mobility, the relative mobility, or RF, is how far into the gel did our proteins move compared to the dye front that moved all the way at the front. So X over Y is what we're going to call our mobility. Okay? And it turns out that the mobility is proportional to the minus the log of the molecular weight. Okay? So if I plot the molecular weight and I plot it on a logarithmic scale, note, log scale. Okay, I'm plotting it logarithmically. What I find is that for proteins of known molecular weight, I can get a straight line. And I can use this, if I want to generate a standard curve. Now, you can see that here. Okay, and so if I measure this molecular weight, what I find, again, is that the mobility is proportional to minus the log of the molecular weight. So instead of putting mobility here, I could just put molecular weight, right? And these would all be a different molecular weight. Okay, this might be 200, and this one might be 100, and this might be 75, and I'll get a beautiful straight line. So now if I don't know what the molecular weight of my protein is, I can run it on an SDS page gel, and I can simply say, oh, well, what was its mobility? Well, its mobility was right here. Okay, and then I simply walk that up to the line, walk it over, and that tells me exactly what the molecular weight was of my unknown. So if on an exam I were to give you a curve like this and tell you that your unknown protein had a certain mobility, you wouldn't have any trouble telling me what its molecular weight is, would you? Most of you, I bet, cheat. Most of you probably use the molecular weight markers on your gel and say, it ran somewhere between this marker and that marker. It's probably about 55 kilodaltons or something. But what I'm telling you is you don't have to do that. You can measure it exactly. And you can say for sure it's 53 or 54 or exactly what it is. Okay. Now, there are two other techniques that we can use to separate proteins by electrophoresis without doing SDS page. Now, by the way, why is it called page? I told you why it was called SDS. What's that page thing stand for? What's this page term mean? Polyacrylamide. Yes. Thank you. Polyacrylamide gel electrophoresis. So now you know what SDS page is. The SDS is sodium dodecyl sulfate, polyacrylamide gel electrophoresis. Very good. Okay. Isoelectric focusing. And that's a very cool technique that separates native proteins. On the basis of relative charge. On the basis of relative, I'm going to change this to content of plus and minus charged amino acids. Okay? So, no SDS. Okay? I'm not going to use SDS at all. Now, you would agree, right, that all proteins are going to have some mix of positively charged amino acids and negatively charged amino acids. And so I want to define a new term. We're going to call this the PI, which is the isoelectric point. Okay, and that's the point, right? It's the pH where the net charge of the protein equals 0. Okay? 0, nothing. Remember then that the isoelectric point, the pi, is going to be some function of the relative amounts of aspartate, actually I'll write single letter code, of aspartate, glutamate, histidine, arginine, and lysine. Okay? Let me give you a concrete example to help this make sense. Let's take two proteins for our example that have different isoelectric points. Okay? So we're going to start with two proteins. Example. Two proteins. Cytochrome C, which I'll draw in orange, which has a pi of 10.6. Okay, it's a pretty basic protein. And the second example that I'll use is our old friend from before, serum albumin, which I'll draw in blue, which has an isoelectric point of 4.8. Now, what does this mean, isoelectric point? Well, let's think this through. What happens if the pH is below 10.6? What's the net charge going to be? Positive, right? pH less than 10.6, this is going to be positively charged. And if the pH is greater than 10.6, negatively charged. Same thing here. If the pH is less than 4.8, then I'm going to start protonating more and more of the aspartate and glutamate groups, and the net charge here is going to be positive. And if the pH is greater than 4.8, it's going to be negative. So the trick here to do isoelectric focusing is I'm going to make a gel with a stable pH gradient in it. Let's not worry for now about how I do it. Let's just say I can do it. So if I have a gel, a long rod gel, not a flat slab, a long cylindrical gel, okay, and this is low pH, and this is high pH, and that's a stable gradient, right? So the pH goes from, I don't know, pick some number, 2 to 10, right? And let's just say, for example, that this is where 4.8 is on the pH range, and that this is where 10.6 is. Well, I'm going to do a little trick here. I'm going to make this the anode down here at the low pH range, and I'm going to connect that to a positive electrode. And I'm going to call this the cathode, and I'm going to connect that to the negative, right? Because cations are positive, cats are positive. They're going to run to the cathode, which is negative. Okay? Anions are negative. They're going to run to the anode, which is positive. Let's think about for a minute what's going to happen. Now, I'm going to have a stable pH gradient, and I spread my protein uniformly through the gel. Uniformly through the gel. So I'm going to take my cytochrome C, and I'm going to let it go everywhere in the gel. Okay? And I'm going to take my albumin, and I'm going to smear it everywhere through the gel. Now, what happens to the cytochrome C molecules that are sitting here, down here? They just happen to find themselves at a pH below 10.6. What's their net charge when they're below 10.6? Positively charged. So which way do they go? They're moving this way. And what about that cytochrome C molecule that happens to find itself just by chance sitting here at a pH that's greater than 10.6? What's its net charge? So where does it go? It goes this way. And how about that molecule that finds itself right there, by chance, sitting at 10.6? Where does it go? Nowhere. Okay? Now let's do the same thing with albumin. Those albumin molecules that find themselves at a lower pH than 4.8, their net charge is, let's say it again, albumin molecules find themselves by chance at a pH less than 4.8. What's their net charge? So which way do they go? This way. And those albumin molecules that have a pH that's greater than 4.8 will be negatively charged, and they will move this way. And the rare molecules that happen to find themselves at a pH of exactly 4.8, where do they go? Nowhere. So what happens over time is all the blue molecules start to accumulate right here. And all the orange molecules start to accumulate right here. And when I'm done running my gel, I end up with a gel that looks like this. End state. All the orange molecules are stacked up at 1 pi. All of the albumin molecules are stacked up at another one. What I've done is I've been able to separate my proteins in the native state, no SDS, and I've separated them based entirely on their isoelectric point. And this technique has very high resolution. By that I mean I can separate proteins that have a difference in pi of 0.01 pH units. So I can really do a pretty good job with this technique. What about two proteins that have the same pi, same isoelectric point but a different molecular weight, or proteins that have the same molecular weight but they have a different pi? Is there a way that I could put these two techniques together? Well, there is, and the approach is called two-dimensional gel electrophoresis. 2D gel electrophoresis. And it involves two steps. Okay? So the first step, the first dimension, is always IEF, isoelectric focusing, with native proteins. No SDS around. Okay? So now again, I'm going to run my gel. I'm going to have this was the positive end that was low pH. This was the negative end that was high pH. And I'm going to run my gel, and I'm going to get a whole bunch of proteins that stack up like this. Okay? Then I'm going to do my second dimension. Okay, so I'm going to now soak the gel in SDS. I'm literally going to take this cylinder, this polyacrylamide cylinder, and throw it in a solution of SDS and let the SDS diffuse in and the proteins unfold, right? And I'm going to, in SDS, and I should say beta-mercaptoethanol. I really want all these proteins to be completely unfolded. And now I'm going to take my old-fashioned SDS page gel, but instead of individual lanes, I'm going to put that whole cylinder up here at the top that had all those lines in it. And now again, I'm going to do my regular SDS page gel. I'm going to put a positive electrode here and a negative electrode here, and they're going to separate here based on the molecular weight. And what I will end up with then is a whole bunch of spots. And I have a picture for you of what a real SDS page gel looks like. That's this. And you can usually, if you're lucky, you can sometimes see more than 1,000 spots. So when you do this, you typically get greater than 1,000 spots. Now, what do you know? Let's pick three proteins. Let's pick an orange protein, and we'll pick a blue protein, and we'll pick a green protein. Okay? What do you know about the orange and the green protein? Would I have been able to separate them on a regular SDS page gel? No, because they have the same molecular weight. But I could separate them by the isoelectric points. What about the orange and blue protein? Could I have told those apart on an IEF gel? No, they had the same PI, but I could see them when I'm separated on a molecular weight gel. So you guys know when you look at a regular SDS page gel, you can see, I don't know, maybe 100 proteins, right? Maybe 100 discrete bands. Hard to see much more than that. This gives me the advantage of both techniques. The IEF gel, which was the first dimension, and the second dimension, which was the SDS page gel. And this technique is remarkably powerful. Okay? And why does it work so well? Because the probability that any two proteins have both the same isoelectric point and the same molecular weight, pretty small. So it's a pretty good technique.